Welcome to Design, Build, and Deliver with Heart Companies, where we dive deep into the world of life science with leaders who decide, design, and deliver the next generation of pharma and biotech facilities. I'm your host, Samir Kondalia. Thanks for joining me as I interview visionary leaders shaping biotech and pharma facilities from C suite executives to project decision makers. Hear real stories, industry trends, and actionable insights to help you build your facilities and your career smarter, faster, and better. Let's continue with Design, Build, and Deliver with Heart Companies. Our guest in this episode is James Dean Vogel, a recognized subject matter expert in bioprocessing and biotechnology with more than 40 years of experience across the biopharmaceutical, food, and cosmetic industries. Jim is the founder and director of the BioProcess Institute, which he launched in 2007 to bring expert testing, training, and consulting together under one roof for the biopharmaceutical industry. Over the years, he's worked in leadership, project management, and engineering roles with companies like Amgen, ESK, Sanofi, Lipton, and Avon, including serving as lead designer for Amgen's Rhode Island facility. Beyond industry, Jim is also deeply committed to education. He is an adjunct professor at the University of Rhode Island and has served as URI's director of the Pharmaceutical Development Institute. He is also heavily involved in industry standards, serving on the ASME Bioprocess Equipment Standard Committee and holds degree from Rutgers University and Manhattan College. I'm excited to dig into his experience, insights, and perspective. So let's dive in with Jim Vogel. Good morning, Jim. Welcome to Design, Build, and Deliver with Heart Companies. Thanks for joining us.
Glad to be here. Thank you.
In our industry, everyone talks about speed to market, first to the market, whether when you build a product, when you build a facility, get to the market. And there are lots of issues that impacts that causes delays. In your opinion, what issues are relevant and important that impact speed to market life science capital projects, and what issues are overblown or misunderstood, which gets a lot of attention but really not that important?
I think it really depends on who is involved with this. So if it is a large multinational, for the most part, they have their capital program set up, work with their facilities and try to do things. And then it really comes down to, do they make a decision, make versus buy? Do they contract or do they go somewhere? And then it comes down to what are their current assets and what's the utilization and what to do. I think that's kind of, they have their own nuances. Each organization has their way to go through that. And unfortunately, it's usually too late because they don't have a plan how to adapt quickly, which I think we'll talk about later in the podcast. As far as the younger companies and so on, I think it's really due to they don't have the staff. So they rely on firms like Hart or other firms to kind of figure out what they need for capital. And they may, in their clinical stages, be going to contract manufacturers or making things more at risk. And then if they go that latter, they're coming up with processes that may not be adaptable to being run in a GMP facility. My example would be they're more hood-based activities such as cell and gene therapies for sure, but also just more laboratory-based because the scale doesn't require it. And then when it comes to scale up and we got to get more into industrial types of equipment, larger chillers, heat transfer, those things, they aren't geared for that. They run a water bath or they run a localized chiller, and then now they're making many larger scale and they now need a chiller plant or they need something like that, which isn't easy to do. I've in my career also had the challenge with, let's say, a biotech company. They like their address, right? So they like where they are and they're like, well, we're going to put it right next to us. Well, a lot of them are in urban settings,
Right.
And now it becomes a retrofit as opposed to a greenfields plant or something. And the costs go through the roof just from because they like, which is great. It's sexy and things, and they might have cash available, but now, all to your point, they have the delays.
By the way, you just described a lot of projects in Cambridge.
Yeah. No, Cambridge, and I was in New York City, and,
Right.
you could go around the world and see this, right?
Right.
And when you're in those areas like Cambridge, historical buildings or whatever. It just complicates things as opposed to, we all know the success is going on in North Carolina right now. You take an old tobacco field and you could pretty much build whatever you want. And I've been involved with many facilities where they first build the Butler building and then they
Mhm.
put the clean rooms inside, where it's a
Mm.
lot easier. You're inside, you're away from the elements, and you can get this done rather quickly, which I think we'll talk about later.
Right. You also mentioned something very interesting. You said process, right? With you, the small companies, they design process, but when you go to scale it up, they don't really fit the manufacturing facility, or it's hard to build a manufacturing facility when you already have a process.
Yeah,
Talk a little bit about that, because I think that's very, very interesting. It's very not easy, but it's very tempting to design process certain way at band scale when you are in the lab,
sure.
but it's very difficult. It can be very difficult if it doesn't fit the GMP manufacturing facility.
Sure. Well, there was this historic cartoon. I'm a biochemical engineer. I teach biochemical engineering at the University of Rhode Island, where they show the scientist and he's with his lab coat inside his office with a small shaker table with a flask. So the cells
Mhm.
are growing in that shaker table. And then out the window, he says, well, we finally completed the scale up. And you look outside and there's a large flask, basically 5 stories tall,
Right.
shaking the cells, right? And so when you deal with a lot of folks who are so enamored with the molecular biology and all of the key processes of that, they make a very successful thing. They can get the drug into the clinic, they show promise and get it to, let's say, phase 1 or phase 2. But now what do we do to make it GMP, to make it reproducible? And us biochemical engineers have a lot of ways to do that in bioreactors and so on. But it's not as, let's say, easy as it is to just take that flask and make a bigger one outside and put that in.
Even if you can, I don't know if it's a good idea, right, to do that for multiple reasons. Let's talk a little bit more about that. What would be the best way to combine both, where you have good science, but you also have a facility that can scale it up and make things at a commercial scale, something you can make, which society needs, and you can sell in the market. Somebody will pay for it. Because often the facility construction, or at least the design, the conceptual design part, starts way earlier than before your process is finalized, at least the
Sure.
clinical process, right? So how do you balance where your facilities can actually, that's a topic we can talk about, that we talked about earlier, flexibility in design. How can you design a facility which has flexibility for whichever process you design? We can start with one modalities, but then we'll expand it for other modalities as well.
So I'll answer that, but let me go back a step because I think the first thing is, does your staff who are coming up with your process, do they have experience or at least knowledge of what the scalable process would be? The larger
Yeah.
companies have that, right? So they
Yes.
have their R&D group, they have their development group, they have their plant, their MSAT, so on. So they have
Right.
that. The smaller companies don't. So
Mm.
usually they have to partner. And if they, let's say, go to a contract manufacturer, and this is what the larger companies do too, they fit their process into the platform. And this is more what we're seeing now. Larger companies have their processing platform. It's one of the major things where we went from having large scale, let's say 1,000-liter bioreactor stainless steel to getting a high, tighter culture in a 2,000-liter single use. And then, the downstream will be tailored to what the molecule is, but they go to do that. This is much different if the smaller company, the biotechnology company, doesn't have that process. Now, they
Mm.
could license in a platform from one of the contract manufacturers. If they have, let's say, a development agreement with one of the larger companies, they could do that. But that's something you got to look at. Now, they go to firms like Hart, and I know you can help them, and there are other firms that can do that.
Yep.
But in all fairness, I'm not sure you have that key process knowledge to get how to go from, let's just say, flask scale to the process equipment. You know the process equipment very well. You've successfully designed many facilities, but their actual process, you're going to need to get more intimate there. And there are ways to do that, but not common in what I've seen.
And also, I'll just add one more thing. There are also lots of process, what we call critical process parameters, that, you talk about. If you have chromatography column, the diameter. If you have
Yeah.
bioreactor, the, bioreactor diameter, impeller size, all. That's a totally different bioengineering, and that needs to be understood when you design the process. The second part of this flexibility in design was that we also see during our conversation lately with pharma companies, is that the science is changing. The days of when you design a facility for MAB and produce MAB for next 20 years are gone, because the pipeline, there is modality versatility. And now the facilities are being designed with flexibility in mind for modalities, not just the science, but modalities. That means different types of molecules, right?
Yep.
How do you design flexibility in it? I'm not talking about just a different size bioreactor. I'm talking about you might have to move the walls, or you might have to have different unit operations like centrifuge or something else. So tell us a little bit about flexibility in design, because I've been fascinated by this subject when I've had chat with a few people lately.
So my first thing is what has to be, and this is our longstanding thing here at the Bioprocess Institute, and I bring this to my students, is the process is king. So the facility needs to support the process. I don't care what your process is. Now, the
Right,
question
right.
is, how do you support what could come in the future? We don't have crystal balls, but I think good engineering means you need to see how you do that. I've seen many facilities basically once they were designed for a product or a modality and they couldn't change.
Hmm.
Large amounts of money wasted, literally bulldozers running into the stainless steel and taking stuff out. It breaks my heart to see that, the amount of time and labor that went in, and couldn't we do something else, maybe even make beer in the plant or something, but it's gone. So
Right.
I think to your core question is, um, I don't think it's that difficult, and the good people are seeing that, especially now in today's world where we are changing faster than we were. To think
Mhm.
of typical capital equipment, right? When we look at, the economics in the university and so on, 25-year life on a piece of equipment. I don't know many pieces of equipment today that make it 25 years.
Yeah.
And, so we got to deal with our accountants. Like right now, I can tell you my computers in my company, they're expense. We don't capitalize them because they're obsolete by the time we plug them in,
Right.
and they'll be replaced.
Yeah, I mean, would you, even today, would you, I mean, I wouldn't feel comfortable using something 25 years old, because there have been so many new generations which are much more accurate, much more efficient, and much more easier to use. So yeah, 25 years, just because something can run for 25 years, it's not a good idea. It's necessarily not a good idea, particularly in our industry. I do feel that there is a way, and this is a real chemical engineer in me, is that there is a way where you can design a facility where you can have a biologics process 101 that will start with cells, then we'll cultivate cells, and we'll purify it, and then we'll isolate the interest of protein, and then drug substance and drug product. Some sort of standard unit operations, and you build a facility which can do scale up, which can do cell cultivation, which can do vaccine, and then it has options. You can combine different columns and membranes. So in one facility you might do map this year and vaccine next year and mRNA the year later. I think it's coming.
Oh, yeah. No, they're here. They're here. I mean, I'm gonna tell you, I've been in many facilities, and been involved with them. They're doing that batch to batch now in
Okay.
in setups in the same room.
Right.
So I think we're there. So let's go back to the core thing. So if you were designing from scratch, the facility needs to be there, and I think one of the core tenets, and so do you taking notes at home, or maybe we could put the graphic up here, is it has to be designed to be renovated. Similar to how, if you look at when you go to large convention centers or hotels, they have walls that can move easily or something to set up for a small
Right.
party or whatever it may be. Now, you may not be able to do that in a night, but the idea I would say is the rule of thumb should be a week or so. You should be able to turn something down, modify it, and get it back up and running. And with proper planning, and there are many people that have done this, you can do that. So you need to do core things when you start with the facility. And again, this is a foreign thing for the smaller companies. The larger ones, for the most part, get it. Things like centralized utilities in a main spine, So then you have shut off valves and everything. And it may cost a little more. And usually we're in this budget create and we'll delete valves or whatever. The point is you say, no, I have to design it because I may turn the south side of the building off to do a renovation, but the north side's still running and it has
Hmm.
to stay going. So
Right.
you design with that in there, and then you bring that to every one of your support utilities. So whether it's clean steam, WFI, or go into the dirty side, so to speak, chillers, house steam, HVAC, you come up with ways to do it. But if you do that properly, and then you can do that. Now, other things that we've employed are things like modular clean rooms. So you can bring in this
Hmm.
modular room to one of those areas that you shut off. So let's just take a very simple design. You have 4 suites, for lack of a better word. You could keep 3 running, turn 1 completely off, knock down whatever walls you have, renovate it, do whatever, and bring it back on.
Hmm.
This is
Right.
all with proper planning and all, but you got to start at the beginning when you design that building. It's designed to do that. And the concept I would use is think of it like whether it's a convention center or maybe a hotel, the product is just a visitor staying in that room for a year or whatever, and then it leaves, and we're bringing another one, and they may need other accommodations. And we
Right.
just do that for them where to go. Obviously, if your process changed drastically, like you go from traditional biologics and now you have solvents, well, then you're going to have to do a H rating or whatever, and that might take a little more than this. But you said I'm going to design the facility with centralized utilities, the ability to turn sections off. Let me timely make these changes and bring it back up. Again, the best in class people are doing this now. They'll shut down for a month and they'll triple the amount of production in their plant by making that change.
Right. I just had this chat with Jack Campion, and he explained there's a flexibility in design concept as Lego, that you have box of
Yeah,
Legos and you
that's a
put
good
the
idea.
pieces together however you want. That doesn't mean you have to throw the rest of the Lego pieces. You use them if you need them next time. And I think that's what anyway. So where does digitalization fits in all of these changes happening? Because digitalization has always been there for like 30 years, right? In some form. It was internet, and then there's pet, and now with AI, machine learning, and then we have these tools that people can use in field. Now they're validated, validation, and all that. Where have you seen meaningful progress in terms of latest facilities
I
or
think
digitalization?
I'm going to say I haven't seen much, but we should be there. We're behind other industries. And so that would be my thing. I believe wireless technologies frees us up for renovations that we don't need that back infrastructure of wired connections. That's a pain in the neck alone to bring out the wires and to do the things that you have to do. Other industries are doing this. And the example that was pointed out to me once is if you look at when the Department of Defense is flying drones around the world, they don't have wires connected to them and it's all encrypted and the data is accurate and they're able to direct their assets however they need to do that. We should be able to take a temperature or a pressure from a vessel in a room with the receiver in the next room and not worry about the wiring, which would free us up from a lot of validation and other things and make
Hmm.
that handshake. They do it
Right.
across the world. Why can't we do it with us? Using the cloud for data and those things, the financial industry is using it. They're doing it and trusting it with all the money that's out there, we should be able to know ones and zeros on the temperature or the pressure of a reactor. But we still seem to be stuck in these, in my opinion, antiquated systems
Hmm.
that we shouldn't go there. It's an industry challenge. I think you see some pockets where we're breaking out of that, but it that's my disappointment. We aren't anywhere near other industries in this aspect.
And also, other side of the coin is that we are in a regulated industry which directly impacts patients' health. So we want to try new technology, but we can't really try it until it's mature and error-proof, whatever
Yeah,
that
and
means.
I disagree with that. The small C in CGMP means you stay current. If you think the financial industry isn't regulated, I think you're wrong.
No,
They
no.
don't want to lose people's money in any way. And, it's a business need too, right? Their credibility goes way down and they can make these exchanges.
I think so. The thing is that, okay, financial industry uses and people use money. Well, it's money. But if we make mistake, people die. But I do
I'm
agree. No,
sorry if it's the same thing. The data needs to be confirmed and there's ways to do it. My thing is I think it's kind of like, we have this automation industrial complex, I think, that's not willing to open up to these other things. I don't buy the regulation side. You could very easily validate wireless transactions and server-based transactions.
right. So what I'm saying is that we can take the same approach that we take with process. We do lots of things in non-GMP at the bench scale in pilot when we reactox material, right? So all these systems can be tried there in non-GMP and tried and tested before they are GMP ready. But we are, I think that's a common criticism of our industry. We are way behind the technology curve when it comes to new technology adoption and adaptations. Lots of already things out there which we haven't really matured and made it part of our day-to-day life.
Well, and I'll go back to FDA has said this. So they had the GMPs for the 21st century. Janet Woodcock went in front of Congress and literally said, I'm paraphrasing her words, is we are making drugs with technology from the 1950s. And this was
Right.
in the 2000s. So, I
Right.
mean, if the head of the FDA has to say something like that, they want this to come in. And
Right.
I just think we have, and I don't want to offend my friends who are making very good livings in this, but I don't buy this regulatory argument. If you have good science and you show that you have a robust capability to show the traceability, you can do this.
Right.
And it's not
Like
just
every
on non-GMP. I'm sorry.
yeah, like everything else. Well, we have process. Now let's put everything we talked about using a real world example. Let's use cell and gene therapy. And I'm talking about cell and gene therapy because cell and gene therapy is, as we talked earlier, that it gets lots of attention. The science gets lots of attention for cell and gene therapy because it's so exciting and it's so groundbreaking, but not the cell and gene therapy manufacturing facilities. Because, as I said, science takes all the attention. Also, cell and gene therapy are very, very expensive. It's like $1 million a shot if they make it to the market. What are your thoughts on the manufacturing strategy for cell and gene therapy? And you can talk about allogenic and autologous cells from different individuals, same individuals, the cost effectiveness, accessibility. But your thoughts in subject, however you want.
So I think we're making strides, but let's face it, the technology has evolved from the hospital labs, the medical center labs, the university labs, right? And
Mm,
the scale is the key thing here. Our batch is 1 to 2 liters.
yeah,
And
it's one shot.
for autologous therapies, it's the same blood, right, that's being processed, and it goes back to the same patient. So the
Mm,
whole idea of batch is a new concept. The regulators, ICH, EU, FDA, have published a lot of good guidance for this. There are licensed therapies now that are out there doing this, so I think we're there. But the process is essentially the same process that the postdoc does transfers to the cells, manipulates them, makes life-saving therapies, but the process isn't stainless steel tanks and pumps moving this. It's very, very small. I could talk about this all day. So my first thing is cutting edge things are companies like Multiply Labs out in California. They're working on robots and even humanoid type robots. Fred Peretti and crew out there are doing a great job of breaking through this. Not
mm,
everybody's going to go there, but that's a way that you can make this scalable right now. Their model was take the actual postdoc process and turn it into something where we get the humans out, less error, and so on. So they're doing really good. Maybe you could put a link to their site in the
yeah, I will. I will.
podcast. The next thing is my vision of where this is going is this will be, and we've always talked about this in biochemical engineering, as I'm going to say since the 90s, is it's going to be like fermenter on a chip that we've been trying to do.
Hmm.
As the automation, as you said, and the technologies get smaller, our process is going to miniaturize. And the entire process will fit on a bench. And my view is someday, I hope I see this, the entire process will be in something like the size of a suitcase. Think of it like a 3D printer. When you asked us years ago that we were gonna have a 3D printer that was gonna print a complex
Right,
we're even doing, turbines for jet engines in 3D printers now and things like that.
right,
Cell and gene therapy, I believe, or cell therapies, I believe, will eventually be, and Fred's, I think, on the cutting edge there at Multiply Labs of doing this, of trying to get this miniaturized. That's where it has to go. Back to the facilities right now is you're going to have to design it for the most part, whether it's a room where your cells are processed and then you'll change it over like a hotel, just like you check into a hotel, stay there for a night and leave, your cells will check in and then they'll
right.
clean it and then they'll leave. And then that'll miniaturize maybe to lab benches or, more RABs or something like that, where your cells will just be in a confined space. And then eventually, like I said, it's going to get to this suitcase, and that suitcase will end up being at the CVS on the corner. So you're going to drop off your dry cleaning and you're going to drop off your cells. You're going to come back next week, pick up your dry cleaning, get your cells, and be cured from whatever disease you have. That's where this will be someday. I hope to see it. I know my grandchildren will definitely see that.
That will be very exciting. By the way, on a side note, the dry cleaner where I drop off my suit is right next to CVS,
There
just
you
to
go. There you go. So there,
so
it's going to
yes, I
work out fine, and maybe you could pick up something else. There'll be, another market there that you can find something else while you're all in one trip.
yeah. I think these are exciting times. The challenges are there, but we
Yep.
have bright people working on it. The problems were never unsolvable scientifically. It's always making them cost effective at scale. That has always been the challenge.
Yeah.
Science has been fascinating. I mean, there are fantastic things that scientists can do in the lab, but to commoditize, that's the problem.
But manual manipulations and aseptic practices, we have proven over and over again, are not reproducible as well as if we can automate it. And let's face it, maybe not for cell therapies, but in processing in general, and this is where Janet Woodcock was going. Continuous processing is something that we can automate. Other industries have been doing this forever. Oil and
Right.
gas, polymers, they've been continuously processing less waste, less a chance of contamination and so on. We still haven't crossed that. I think upstream will be a little difficult. You can still do semi-batch or whatever for cell growth and so on. Downstream, there's next to no reason. We're making improvements. You're seeing things like online dilution of buffers and those things where they're becoming more and more common. I think column steps and other things like that can definitely be more, streamlined, but it's back to all the things you said, the automation, along with knowing your process and making that happen and showing that it's reproducible. Some of these platforms that I mentioned earlier in the podcast are incorporating them in specific steps, but we're still not to the point. There's this graphic I use in my training where, you dump basically chemicals into one side and pills come out the other side in a magic black box that has happened. We're not quite there. There have been some people that have built machines that kind of get there. FDA has sponsored them. Some of the universities have been doing things. But I think if you take groups of unit operations, we can definitely do that.
The other issue I think is that the timing too. The things that you mentioned, they're definitely doable, but they will take time. They will take years to have the strategy and system in place and all these things. The leaders, our leaders, they are focused either on next election or next quarter. Not a criticism, it's an observation. That's where all the focus is, right? So it's very hard to find someone who is motivated enough, who will put something in place which will not come to fruition long after they are out of the office. But that's a separate topic, and we need a separate conversation for that. But I think that's Saminger's issue too.
I think the key things that we talked about. So flexible facilities are possible. It does need some planning. I think if you get the right folks involved, I know Hard has those people. And we try to do it with our clients if we can. One specific thing I would say is take your preliminary design and bounce it off of a new set of eyes. Make it a day or something. If you could get 2 or 3 people in the room, that usually helps. And then you go back to your designers and you say, well, here's some comments we heard. It might be simple as move a door here and we'll get this much more throughput. Or it may be fundamentally, have you looked at a company like Multiply Labs to do this technology? You might want to think someday that will come in. How do we include it?
Hmm.
that would be one tangible thing, I think, going back to the main theme of this podcast of flexible design. Get another set of eyes up front. It's a lot cheaper to change it on paper than to do it with, bricks and mortar or stainless steel or whatever you have.
That's exactly what Jimmy O'Keefe said. I interviewed him for one of my earlier episodes, and he said it's a lot easier to make changes with a click of mouse on a computer
Yep.
than take a wall out.
Yep.
Jim, outside of work, a little bit personal about yourself, what are your favorite books, TV shows, movies that you do to relax and something you enjoy? Music?
I'm mostly nonfiction person on the books. I just read the story about Bruce Springsteen and making the album Nebraska. There's a Netflix film version of that.
Oh, yeah, you're from New Jersey. So
I am from New Jersey, and so yes, Bruce Springsteen is right up there on the mountain in our view from people from New Jersey. So I am
right.
there. We play all his songs when I go home to my Rutgers football games and things like that. I ride my bicycle, and here in Rhode Island, we have a beautiful place, and we're on a river that goes to the ocean, and so do things like that. I'm very blessed to have 3 grandchildren, spend time with them, and do that. As I'm getting on in my career, I really enjoy teaching at the University of Rhode Island with the chemical engineering and the biochemical engineering program. We're establishing that, getting it going, and just interacting with the students to see them. My goal is strictly, I want them to come out and be productive in this great field. I have a passion for this. I've had it my whole career from the first day that I heard how you could get these growing entities to make your product. I've been involved in biochemical engineering ever since, and I want to be here and continue doing that and pass it on to the next round of biochemical engineers.
That's great. Jim, if someone wants to get in touch with you, if they have any questions or want to collaborate or want to ask questions about something you said on the podcast, what's the easiest or your preferable way for them to get in touch with you?
You can contact us through our website at bioprocessinstitute.com. I'm sure you'll show it there. We have an info ad or whatever. I'm on LinkedIn if you needed to do that. Or you can give us a call here at the office. I'd be more than glad to help or whatever we can do there. We support our clients here. We do some testing, consulting, and training here at the Bioprocess Institute. And we're here to help where we can and add value where we can.
Thank you very much, Jim. I'll put all of that in show notes, and I am looking forward to visiting your office as we talked about earlier.
Well, you're close by. We're looking forward to it here too.
Yeah, we are not too far from each other. Jim, this has been fascinating and very interesting. Thank you very much for your time and sharing everything you shared with us. Thanks again. Thanks a lot. And you have a wonderful day, Jim.
You too. Have a great day. Bye-bye.
Thanks for joining us on Design, Build, and Deliver with Heart Companies, where we explore the people, projects, and ideas shaping the future of life science facilities. If you enjoyed this conversation, be sure to follow or subscribe so you don't miss upcoming episodes with more leaders from across the biotech and pharma world. To learn more about Heart Companies, our integrated design, engineering, and passivation services, visit heartcompanies.com, that is HART companies.com, or connect with us on LinkedIn. I'm Samir Gondalia. On behalf of everyone at Heart, thanks for listening. Until next time, build with purpose, deliver with pride.